Can you see the little recording light?
Oh yeah, I think I can, yeah. Yep.
Great. Okay, so we're recording.
So, hi, this is Raja. So, Sue, I will start exactly at noon, like, or you want to wait a minute or so, and then after that, Jonathon will introduce Biocompute, and we'll go from there.
That sounds good. I mean, if you want to give people another minute or so to wander in, but we're up to 18. I think there were, what, 24 on the sign up? So we could probably start the introductions and let people meander in a minute or two late.
All right, I think, yeah, so that's good. Yeah, because I don't want to take time away from the rest of the stuff. So
Yeah.
welcome everybody. And thank you, Sue, and thank you, ISB, for organizing this. I'm very excited, we're very excited to talk about Biocompute, and especially how Biocompute can be used for biocuration, and how we have been using it in a couple of our projects. And just like any project which evolves over time, Biocompute has been evolving for a long time. Initially, it was developed for just recording workflows, but now it has evolved to also help in organizing data in knowledge bases, for example. So today, Jonathon Keeney, who is going to do the first introduction of Biocompute, is going to provide a background on what is Biocompute, why did we develop it, how was it developed, who are the players in developing it. Then the next one is Rahi Navalkar. She works for GlyGen, which is a database project, glycoinformatics resource. She's going to say or talk about how Biocompute objects are helping her as a curator to organize data and keep track of things. And then in the front end, how that Biocompute can help in evidence tracking. Now, that's an implementation. So we're going to talk about implementation first before we go into an example, how to use the Biocompute object editor. And Janisha, who is from the Biocompute team, is going to talk about it. And then Hadley, who has been working closely with the Galaxy community, will talk about how he has been working with them to integrate Biocompute editing and creation within the Galaxy platform. With that, Jonathon, all yours.
Oops, there we go. Okay. Am I unmuted? Okay, great. Okay, thanks a lot, Raja. Let me just minimize presenter tools here. All right, there we go. Okay, so let me echo what Raja said and say welcome. It's really cool that we still got to do this workshop, even though the original in-person conference obviously couldn't be held. So thanks a lot for showing an interest in participating in the virtual session. So it's split into two parts, and I think Raja already covered the first part pretty well. The second part, again, just to sort of echo what Hadley said, we've got breakout sessions. These are three concurrent sessions. Each one has its own kind of focus. So there will be an overview in general with sort of strategic and programmatic goals that Raja and I will be hosting. Hadley will host a session on technical and computational considerations, can talk about the language and implementations and things like that. And then Rahi and Janisha will be available for talking about the use of Biocompute in knowledge bases. Okay. So the original seed of the idea for Biocompute goes back to high throughput sequencing data flows, or HTS data flows. And so in HTS data flows, you'll typically get a very large volume of data, which is represented by the reads on the left-hand side of the slides that are coming off of a sequencer. So there'll be this enormous volume of data, and that can be processed in any one of a large number of ways. You might do ancestry or cancer detection or microbiome, disease correlation, etc. A lot of times you can analyze this, it may be the exact same data, maybe even the exact same platform, possibly even the exact same tools, and you can get very different analyses. And small changes in data processing can get really amplified when you're working with big data. And so oftentimes you might spend a very long time building and tweaking and refining a pipeline, going back and testing it, maybe putting it through some paces with some synthetic data to see really what it can do. And then once you finally harden that pipeline, it could be 6 months that have gone by and you're really, really, really deep into the pipeline. You understand it very well. And it becomes very difficult to explain to somebody else what you did who was not along for your thought process and doesn't understand. And so it can very much seem like a black box to someone who's not familiar with it. And so when you go to try and explain this to someone else, there's all kinds of data that's frequently missing. It's very similar to what you might see in a wet lab environment. You could not just give someone the final image, for example, and expect that they would be able to interpret that. You would need a lot of information to come with that. What is it that you were trying to do in the first place? What is, if it's a fluorescence image like this, what's the cell type? What are you marking? These kinds of things. And then even more granularly, if you wanted to reproduce that, what's the strain of the animal? What's the tissue type? Maybe what's the lot number of the fluorescence indicator, things like that. So you need a lot of information in order to make sense of any of those. And computational biology is no different. You need the inputs, the parameters, the execution environments, the outputs, etc. And so this was the thinking that Raja Mazumder and Vahan Simonyan had back in 2012, and they worked very hard to convince others of. Everyone, frequently kind of has a different idea of what they think is important to record. And if they don't record it, it can be a really big pain to track that person down and get that information from them, especially if they're not really in the same state of mind as they were back when they recorded that pipeline. And so this has resulted in some inefficiencies all over the place. One of the more notable ones is in the reproducibility crisis in academic peer review publishing. But probably one of the most concrete places where it's been observed is in formal regulatory submissions to the FDA. And so in a situation like this, an industry sponsor will submit something to the FDA. Frequently, we're told what happens is the FDA reviewer will have to kick it back with some sort of clarification on something. And the industry sponsor then has to sort of interpret those comments and figure out what it was that wasn't clear. Because this is not a situation where you can just sort of sit down next to somebody at their computer and kind of walk them through it. You have to draft very formal responses. And so they have to figure out what it was that was not clear and try to draft a response to that and resubmit it. And maybe they'll get it right on the second time and maybe they won't. Sometimes they'll go around many times just to be able to get the FDA reviewer enough information to make a decision. And so this is a huge waste of time and money. It's been estimated in the millions of dollars and months of time just to make a decision. And so in 2014, the Genomics Working Group at the FDA, which is a cross-FDA working group, held a special session to try and address this topic. And they produced an internal document that's summarized by these 4 points. Number 1, any solution to this problem should be human readable, kind of like a GenBank sequence record. Number 2, it should be machine readable so that you can work with those fields in some way. So there's predictable structure to it. Number 3, it should contain enough information to understand the computational pipelines, interpret that information, maintain records, and reproduce the experiments. This is kind of the core of the entire idea. And then finally, it should be immutable. So you can make sure that you know that the information hasn't been altered. And so the solution is what we now call Biocompute. So Biocompute is an IEEE approved standard for communicating bioinformatic analysis workflows. So you notice it's no longer HTS. It's since been broadened to many other things, including modeling and simulation, even out beyond the life sciences. But one of those things that it's probably had the most testing with is use in knowledge bases and biocuration, which is what we'll talk about today. So it acts like an envelope for an entire pipeline. So from beginning to end, what did you do? How is the data transformed? What other considerations are there? It was written to be human and machine readable. So it's written in JSON, which is JavaScript object notation. This is not a programming language. It's just a way of organizing and structuring data. It's categorized by domains. So all of the information is conceptually partitioned into these meaningful categories. So if you want to know what the execution environment was, you would go to the execution domain. If you want to know what the parameters were, you'd go to the parametric domain and so forth. So it's meant to be intuitive and easy to understand. And since it's written in JSON, you can kind of build this in whatever presentation format you want. We'll show you an example of a way that, that we've done it using what's called the tree view, so you can collapse the stuff you're not interested in. But these fields are internally referenced. So if you wanted to, you know, maybe associate the parameters with any individual parameter with any individual step, you could very easily do that because of the way that the schema is written. It adheres to and encourages FAIR principles. That's findable, accessible, interoperable, and reusable. So we've had a few folks on as part of the consortium that are very, very strong FAIR advocates and have made sure that it adheres to those principles at every turn. One of the ways in which it does so is that it's maintained as a fully open source repository. It's very adaptable, for example, to other schemas. We've actually included 3 other schemas in ours. And it's also the fact that it's maintained in an open source repository means that you could fork off your own branch of this and work with it yourself. And it's also got what's called an execution domain, excuse me, an extension domain, which I'll talk about in just a couple of slides. So there's a lot of ways in which it's adaptable. It preserves data provenance, so you'll be able to see with very great detail how the data is transformed at every single step, which is very nice for tracking. It's also got a very strong user attribution. So it's got a very specific curated language for user attribution, so you know exactly what each person did along the way. So it uses provenance, ontology, and versioning, which is Prov-O. And then finally, it's got unique IDs for versioning in case you need to version one of the pipelines. So this is a sort of a very, very high level view of what a BCO might look like. This is a smaller one, and you're not expected to read the JSON that's in there. It's just to kind of show you the ways in which it's all partitioned. So there's 8 top-level domains. 5 are required and 3 are optional. So there's a provenance domain for metadata, description domain, which describes all the steps in the pipeline and gives a description of them, an error domain for kind of demarcating the bounds of the pipeline, so false positives, false negatives, you know, whatever is appropriate for your pipeline. A parametric domain for recording any parameters, an execution domain, which describes the execution environment that it was run in, an IO domain for input and output files, which acts like a data manifest. So the files are not kept on board with this. It's a very small file, but it acts like a data manifest for all those files. An extension domain, so this is the user-defined domain in case there's something in here that doesn't cover your needs. This is the way to be able to add that in, whatever it is that you need, and still be compliant with the standard. And then finally, a usability domain, which kind of functions like an abstract for the entire thing. You know, what was the purpose of the pipeline? What were you hoping to accomplish? You know, any relevant details, for example, and that kind of a thing. So we've worked with a whole lot of people over the years since the initial funding and the initial seed of the idea for the project in public, private, and academic sectors. I won't go over all of them. The only one I will mention real briefly is Google. So Google Cloud has mentioned that they will support Biocompute in the Google Cloud project as part of their participation in the STRIDES initiative to host publicly funded data. So that was really exciting for us. I mentioned that it has been standardized. So this is the link to the official standard. The IEEE also maintains, or I should say, I guess, administers the open source repository, which I'll show you in just a moment as well. So it's officially known as 2791-2020, IEEE 2791-2020, which is kind of a mouthful though, so we still call it by Biocompute. The FDA has also published support for it. So the FDA has said that they will accept Biocompute as part of submissions for 4 different new drug applications submitted to any of the 3 participating centers, which is CBER, CEDAR, and CIFSAN. So those are 3 different centers within the FDA. And that Biocompute is now part of the data standards catalog at FDA. So that was really exciting news for us. So that's now in place too. So the Biocompute schema is kept, as I mentioned, in an open source repository on GitLab. I've included the URL to it up here in case you'd like to go check it out. If you do, the top level file that you would start with is called 2791object.json. The screenshot is cut off at the bottom here below license. But what you'll see if you go there is that all the other domains have their own files there. And these are all cross-referenced. So you could go down and pick out any one of those that you're interested in to learn more about it and see what the schema says. So some of the key features of a BCO. So BCO, I don't think I mentioned this earlier. So when you record a specific pipeline in a format that conforms to the Biocompute specification, that's called a Biocompute object, or a BCO. So the key features of having a BCO are, number one, that it abstracts away whatever your workflow is based on some commonalities. So you have some input, you're transforming it according to some parameters, and you get some output, and all of that happens in some environment. And so for that reason, it's very appropriate for lots of different computational workflows, not just high throughput sequencing. It has a usability domain. This is a free text description where you can describe whatever it is that you feel is appropriate. It has a data provenance. It has very strong data provenance. So I said this kind of acts like a data manifest where you can track files from beginning to end. It also has very strong user attribution, which uses a very heavily curated language, so you know exactly what someone's role in that pipeline was. It has what we call a verification kit, which is kind of a quick sanity check. Is the output of this valid given what the input files were and what you're claiming the error was? So you take some set of input files and you are aware of whatever the error level in this file is. And then it's kind of a quick way of just checking to make sure that whatever the output is valid, that this pipeline is even capable of getting that. And then finally, it's very extensible. So I mentioned the open source repository. The extension domain is where you can essentially define your own schema for whatever portion you need and in whatever way you want. And then you can add your own user-defined domain into that and still be compliant with the standard. So applying these to biocuration, I mentioned the number one important part is the fact that workflows are abstracted. So you have some input rather. You want to transform that data according to some parameters to get some output, and all of that happens within some environment. And you may want to add some annotation to that to let people know what you're doing. So that's a very, very generalized way of describing a pipeline. And so it's become very clear that this is a great way to solve a lot of different problems, not just high throughput sequencing. And so we've tried several others. I think I mentioned some of the other ones like modeling and simulation and so forth. And it turns out it works out very well for biocuration too. And so we've seen it used in a couple of different contexts there. It has very strong data provenance and user attribution. These are built right into the BCO. And then finally, the fact that it is extensible. So if there's something that's not already built into the base schema, it's really easy to add. In fact, it's built to be extensible in that way. So it works really well. And so the advantages of doing this are that you can then work with that data programmatically and track down exactly what was done, or track down errors and rapidly update an entire pipeline that could be very easily scaled, for example. You know exactly what kind of data to expect and exactly what format it'll be in. And small changes can be easily, or big changes can be easily made and disseminated very quickly and easily without any confusion. It's very good for teams of people. So one of the previous folks that came and spoke at one of the Biocompute workshops was a Bill and Melinda Gates funded researcher that was working with the World Health Organization on the ReSeq TB pipeline, which is for detecting tuberculosis. And so they have a very big team that's geographically distributed, and they all need to be on the same page, and they really have no room for error. And so he came and gave a talk both to us and to the World Health Organization about how Biocompute is the perfect solution for that. So Biocompute is really good for teams of people that all need to be doing the same thing, especially if that team is geographically distributed. And then finally, flexibility. So BCO can help to standardize a workflow description, but still give you the ability to do whatever it is that that's unique to your workflow. So it doesn't mandate that you use some particular kind of workflow or some particular platform or some particular tool. It's really meant to be kind of this lingua franca of data processing. So that's kind of a nice segue into using Biocompute for data curation and for knowledge bases. And so we'll spend a few minutes just kind of showing some of the tools that we've got. Well, first I'll pass it to Rahi Navalkar, who will go over actually using it, and then Hadley and Janish will talk very briefly about some of the tools that we've developed, just so you're aware of them. This won't be a deep dive or a tutorial, but just to let you know what kind of tools do exist. And this is some of the contact information. So before I pass it off to Rahi, is there any questions for me? Okay.
If there are no questions, I did want to chime in with just a quick comment. I'm keeping an eye on hands being raised, and I'm sure Sue is too. So, you know, at any time, please hit the raise your hand button, or feel free to unmute yourself if you'd like and ask a question. We want this to be as interactive as possible. Also, Sue has been, and I think I'm going to put a couple of links into the Slack space for this workshop so that you'll have access to those as they come up.
Okay. So I will stop sharing now, and Rahi can start sharing hers.
Sure. I just want to check you can hear me well.
Yep, I can hear you.
Okay, perfect. I'm just sharing my screen. Okay. So hi everyone, I'm Rahi Navalkar, and I work on a bioinformatics database called GlyGen, which I will introduce shortly. But the focus today is that I'll be talking about how GlyGen has used BCO in the integration process throughout. So firstly, I want to start by introducing the GlyGen project. So GlyGen is a NIH-funded glycoinformatics project where we have collaborated with several different major resources and have integrated diverse glycoconjugate data. So you can access the GlyGen portal through the link that I've provided here, or if you like, you can scan the barcode and it will take you to the site directly. Okay. So this slide shows the GlyGen data model. And as you can see, we have two main, what you can call as entities, that is glycan and protein. And then they branch out to their individual properties, let's say like function, mass, etc. So GlyGen basically allows users to perform search queries across different domains. And an example of that would be if a user wants to search for a protein implicated in a disease and want to also find out what are the type of glycans that can be found of a particular mass. So GlyGen can allow users to do certain type of complex query under one portal. So how has GlyGen been using the Biocompute objects? So as I mentioned earlier, GlyGen has a large amount of diverse data. So currently we have over 350 individual data sets that we have collected from major resources, let's say from UniProt KB, UniCarb, and GlyConnect. What has GlyGen done is that GlyGen has assigned each of these individual data sets with a Biocompute object, which document basically its integration details. So you can access, again, the entire GlyGen data collection and its corresponding BCOs through the link. So how has Biocompute object basically helped GlyGen, especially during the curation and the integration processes? So to present that, I wanted to highlight one particular use case. That is when we received a data set from a university lab. They had recently published a paper, and they provided us a data set which document for over 300 human proteins and had identified O-glycosylation site. So we received this data via email. So in this particular case, we used BCO to document the author name, the source affiliation, as well as the format and the description of the table and its usability, that what does this table can provide its users. So additionally, what we did was we spoke with the author, and through back and forth email conversation, we decided to add another column of an additional annotation. Then we also performed different type of QC steps, that is like checking the UniProt KBX session, validating the site on FASTA sequence, etc. So in this case, what BCO really helped us was to capture the author's note, for example, like what did author mention about this added annotation, which was not in the author's original table. Then BCO also helped us capture the QC steps that we performed. Then through BCO, a user or the author can access and download all the files because we provide the URLs to do so. So this is an example of how a data set, one single data set, looks in GlyGen. So at the top, you can see it is identified by a unique stable ID. Then each data set is also versioned to track changes. And on the right side, you can see we provide a button associated to each data set where you can access its own BCO in text or in JSON format. So here I wanted to highlight certain sections of that BCO object. This is in a JSON format of what it actually looks like and what does it store. So at the first section, you can see it has a stable versioned URL, and it also has the GlyGen ID associated to that data set. In the next section, you can see it has captured the metadata of that table, such as the name, license, the author's author name, contribution, name, and the role of that person. Then in the next section, the BCO has stored the description of that data set, that is the usability. That is, for example, this particular data set talks about a list of human proteins with information on glycosylation sites and associated glycans from Unicarb KP data. Next, a BCO has the capability to even store the QC steps that we designed. So this particular data set, we had to design a very complex QC workflow, and BCO provided a space to store this information as well. And in the last section, I wanted to highlight how a user or anyone who can access this BCO can use the URLs provided and can download all the files that were involved in producing the final output file. That is, the user can access the input file, that is what was submitted by the author, then the script that processed it, and finally the output file after the QC steps and everything was done. So here, to summarize, I wanted to highlight some of the advantages of using a BCO in a knowledge base like GlyGen. So I think first of all, it provides complete transparency of the workflow to its author, collaborators, and users, of what we have done with that data set. Secondly, it allows us to provide detailed documentation and maintenance of large number of diverse data sets in a very standardized format. Next, because BCO also stores the links where users can download all the files, that workflow can be easily reproduced through the details provided in the BCO. Then BCO also provides a stable version URL, which you can use to download or share it. And finally, because it's in a JSON format, in a standardized JSON format, you can parse certain sections. Like for example, GlyGen has parsed the usability domain and used it as a description for each individual data sets. So I wanted to quickly also go over how do we create BCOs. I'll be showing a live demo in the breakout session, but this is just an overview. So Biocompute objects can be generated using the Biocompute editor. So what GlyGen does is it uses an instance of a Biocompute editor where all the GlyGen BCOs are created and stored and given a unique ID. Now, similar to GlyGen, if any other database wants to create their own instance, they can do that, and the documentation can be found through the link. So this screenshot basically shows the instance of the BCO editor that I was talking about. This is particularly for use of GlyGen, and all the BCO objects with the IDs are stored here. And if you click on the right object, there's create object, it takes you to a form-based like system where there are predefined values which can be used to fill up all the details. So once you press the Save Changes button, then this form is then converted to a standardized BCO in a JSON format. And then because all of these BCOs have an ID, you can search them through that, or the name, or even the contributor that has added the information, the BCO. So once the BCO in the JSON format is created, you can either edit it and go back and make additional changes. And additionally, all the IDs are stored under a particular registered prefix. For example, for GlyGen, we're using the prefix GLY. So similarly, I just wanted to highlight another BCO in a different bioinformatic cancer database called as OncoMX. Similar to GlyGen, the data set captures details for a particular database, and it has its own ID and prefix. So with that, I want to conclude my presentation, and if there are any questions, you can either ask right now or put them in the chat, or you can join the breakout session number 3, where we'll go in detail. Thank you. So next, I would like to pass it on to Janisha.
Hey everyone. Okay, I'm just going to get this into the slideshow view. Give me just one moment. Okay, are you guys able to see this?
Yep, can see it just fine.
Great, okay. Hey there everyone, my name is Janisha Patel, and I'll be introducing you to developing BCOs in cloud-based computing environments, specifically Hive. Sorry guys, would you guys give me just a moment? Hadley, would you like to discuss Galaxy BCO first, and then I can go with Hive? I seem to be having some technical difficulties.
Sure.
Sorry about that.
Okay, so I am going to be trying to do a live demo. So hopefully everything works out. Bear with me. So what I have, I will be discussing the development process for Biocompute. The development associated with implementation of Biocompute objects using Galaxy as an example, but I'm also going to show off our community interaction via GitHub and Gitter. So I'm going to start with that first. I don't know if everybody is familiar with Gitter. It is a chat platform that is associated with GitHub. It's published by GitLab. Sorry, so it's associated with GitLab, but can integrate with GitHub as well as a lot of other development systems. So we have a few different spaces set up under the Biocompute Objects organization. The most generalized one is just Biocompute Objects /community. And this is a space for anybody to post any messages or any kind of chats. We do have a couple of other specific ones. And you're seeing on the left, you know, obviously I'm part of the Galaxy development platform because we've done some of that, but, you know, it can interact with GitHub so that folks can go straight from GitHub to Gitter and communicate the issues and stuff. So in the Biocompute organization on GitHub, we have a bunch of different repositories. There are a few that we highlight. The first one is the BCO specification, which is the repository to support the IEEE 2791/2020 standard. Essentially, this is our space for documentation. Let's see, we have the link here for Gitter to chat, introductions and examples here. The user guide is also here, where we have specific documentation and examples for each one of the fields or each one of the domains. So this is where you can find information about each of those. We also have a GitHub repository for our BCO portal, which is titled BCO Editor in GitHub. And again, these are all open source and completely available for anybody to look at. And contribution is welcomed in the form of pull requests at any time. So for those of you familiar with that, here in the portal, we list general requirements, and we have specific instructions for implementation of the portal. For we have a CentOS and an Ubuntu implementation set available. Like Rahi showed in her previous presentation, there are different implementations of this tool, or you can use our main portal here at portal.aws.biochemistry.edu. So there is also a few other repositories we have here for support of Biocompute objects. So feel free to take a look. Our Galaxy instance was forked over from the main Galaxy repository, and a few of us who work on the Biocompute project modified the Galaxy code and created our own instance of Galaxy, which, by the way, this has been merged back into the main Galaxy branch, and with the release for this month, Galaxy version 20.9, the Biocompute functionality is available in any instance of Galaxy. So our Galaxy instance is available at galaxy.aws.biochemistry.gwu.edu. The long little bit here is from our university, but it's consistent across all of our resources. So if you remember it once, you don't have to remember it more than that. So our Galaxy instance here, this is the main page of Galaxy. The way that we implemented Biocompute objects into Galaxy was via the workflows. So this is a function in Galaxy where you can construct, I'm going to go to the edit here. You can construct a workflow by, over here, you can add a tool, and then you select the data type, and then you can connect it. And you can see here, Galaxy has it built in that only specific tools can link with another one based on the type of inputs and outputs and all of that. So I have a pre-constructed one here that I've used for testing and development. Essentially, it takes a single input, which is a text file, runs it through a tool that downloads a genome from NCBI based on that input. So the input would be an accession. The invocation that I'm going to show you folks is for the SARS genome. Viral genomes are small and good for testing because they can be run multiple times quickly. So downloads the file. We have another program called ART, which will take that reference and generate a set of in silico paired end reads. So you take a genome, you generate in silico reads, then you compare the generated fake reads to the genome and get a result. I mean, it's really nonsensical, but good for, nonsensical in a scientific sense, but good for testing in a computational sense. So one of the key features of workflows is that you're able to, like I said, connect the different pieces, but there's also a lot of different annotation steps that you can add. So here you can see I've included some tags, and I've included a specific annotation for this workflow. It's versioned, so every time you add a feature or change the annotation, the workflow is versioned and it's named. So I'm going to go back to my home screen, and you can see, clicking on the workflow, again, it takes us back to the workflow list. If you select run the workflow, you'll get pre-populated pipeline based on your based on the workflow. And I am going to run this now. Let's see, fingers crossed. Okay, the first job worked. That means usually that the rest of them will work. So right now, it generated the in silico reads. Now it's running FastQ groomer to remove low quality sequences. Then it mapped it via BWA. So you can see, if we go, now I'll show you how to actually get the Biocompute object. Going to the invocations under user, we have workflow invocations. So I showed you workflows, collection of tools put together so that you can reuse that same analysis on maybe multiple sequences or, you know, different times. Invocation is a specific instance of a workflow. So you can see here, list of invocations. I have a bunch of them here, and a lot of them are tests because I'm on my test account. But you can see the one we ran a minute ago is listed here. So expanding this will give you options to see more information about the invocation. Each invocation has a specific identifier that is unique to that invocation on that instance of Galaxy. So by taking the invocation and appending that to the URL, you can see unique, and you can get the invocation. So we have a download Biocompute Objects button here. And so that downloaded an instance of our invocation. Let's see. Sorry about this. And downloads. So just downloaded this one. So this is a raw JSON file. You know, Jonathan walked you through this when he was talking about it in the beginning. I have a add-on in my Chrome that makes it a little easier to read these text files. So that's what I just opened it in. But you can see the URL here. It is a file on my local machine. And then this is the Biocompute object that was generated for the workflow I just ran. You can see the time 9:24, 14:11:16. So here you can see in the keywords, we pulled out information from the tags that were added to the workflow. So in Galaxy, adding tags to your workflow will associate those as keywords for the Biocompute object. Each of the pipeline steps are listed here. In the description domain, we have, there's some portions that are still incomplete. This is not a full ready to go Biocompute object because that's very difficult to do programmatically without any human interaction. But this does get you most of the way there. Let's see, so we have our input and output subdomains. And if you notice, the URIs associated with each of these. So we have, I'll show this to you using the description domain because that makes a little more sense sometimes. In the pipeline steps, we'll look at step number 2, which was Art Illumina. So this was the analysis that took a NCBI accession and generated output forward and output reverse reads. So clicking on this link opens a new tab, and you see this is now not just on my local machine. This is actually taking us to the file listed in our Galaxy instance. And so that was, so this is a lot of information about a file that was really, this is it. But that was our input file. And you can see the output reverse, again, a lot of metadata associated with the file. But you can get to the preview section and see that it was just included in the, here we go. That's what I was looking for. No, that's not what I was looking for. Apologies. Well, that's the metadata for the output file there. So our future plans are to enable, future plans meaning with the next release of Galaxy, which is January, will be 21.1, because Galaxy goes by the year and then by the month, and they do it quarterly. So 21.1 will be the next Galaxy release. We hope to have approved code to, instead of just downloading the Biocompute object, there will also be a button here that will automatically submit this to our portal, which, give me a second here for this to load. Maybe a couple of seconds. Okay, like I said, my internet connection has been a little wonky lately, so I had all the Galaxy stuff preloaded except for running the workflow. So we'll come back to that later, but we do, so. All right, in closing, so this link takes us to, these are all of the resources we have developed for Biocompute or aiding in the creation or use of Biocompute. And they are all being hosted on AWS. That's the time being, thanks to an SF grant, which I'll show you info about in the acknowledgements. I think with that, are there any questions? And I will say, for my breakout session, I would like to, I'll discuss anything you want. We can talk about the weather or anything. But I was planning on discussing GitHub, Gitter, and how somebody could interact or help contribute to the development of Biocompute. It's not going to be, this is how you code this line kind of a thing. Unless there's somebody who wants that. Okay, I think it's your turn now, Janisha.
Okay.
Are you ready to go, or?
Yes, I'm all set up.
Okay, great. Let me, I move things around here. Okay, great, thank you.
No problem. Okay, so hi again. Thank you for your patience. My name is Janisha Patel, and I'll be introducing you to developing BCOs in cloud-based computing environments, specifically Hive. There are several cloud-based tools that Hadley mentioned developed to create Biocompute objects. These tools range from being platform-specific, such as Hive and Galaxy, to platform-independent, such as the BCO editor in the portal. This short presentation will delve into the BCO functionality in Hive. Hive is a bioinformatics platform developed by GW and the FDA and has been used agency-wide for reviews and research. As Jonathon mentioned, Biocompute objects are a record of any computational analysis executed containing all the necessary information to repeat an entire pipeline from start to finish. and includes additional metadata to identify provenance and usage. For computational analysis, a lot of work goes into developing the analysis, adjusting parameters, testing the pipeline, QCing the results. And for a high quality analysis, checking these results can take a lot of time. Often the communication of these pipelines loses some details, leading an analysis to look like a black box to anyone who's unfamiliar with it. Proper computational analysis should contain every step as well as standardized descriptions of the analysis pipeline. And the BCO app in Hive allows users to capture every computational detail from start to completion of an analysis. And so developing BCOs in these cloud-based computational environments make bioinformatics analysis more reproducible and well documented. The decreasing cost, increasing speed, and improved accuracy of NGS over the last decade has led to significant advances in genomic technology. With the sheer scale of data and subsequent analysis, the challenges associated with storing, accessing, and computing on NGS data create bottlenecks for downstream analysis, in addition to concerns about reproducibility. Hive, the high-performance integrated virtual environment, provides web access for authorized users to deposit, retrieve, store, annotate, and compute on NGS data. Now, I'm going to give a demo on how to use Hive and how to develop BCOs. Bear with me for just a moment when I pull up my Chrome. Okay, Hadley, are you guys able to see this?
Yes, we can see it.
Okay, wonderful. Okay, so first we need to log in. Here we have the Hive dashboard. As you can see on the left-hand side, we have the Hive space, which is a place to create folders for yourself, and these are solely yours unless you share. All objects host everything, all personal uploads, and whatever is shared with you by another user, whether it's a genome file, a computation, an annotation, and these are all subsections within, there are subsections within all objects, and these are reads, genomes, annotations, files, computations, and you can selectively search all shared or personal items here in the search box. Inbox contains only your uploads and computations derived from them. These do not include files shared with you. And each table contains 6 columns. There's a Hive unique identifier, a logo representing the type of computation or process run or file, and then a name for the process, the created information, and so on. You just have to, work with sizing. Right now my monitor won't look that great, so I won't bother. To access the tools in Hive, you click Portal here from the menu bar. There's a variety of tools, including annotations, classifications, glycomics, metagenomics, post alignment, and genome sequence alignment. These are just to name a few classifications. There are more than this. So to show a few tools native to the Hive platform, we have Hive Hexagon, which is here, the DNA sequence alignment right here. Hive Hexagon is a massively parallel alignment algorithm specifically designed to work with NGS alignments to reference genomes. We have Hive Heptagon, which is a sequence profiling, which is a SNP profiling tool, and that performs base calling and SNP calling and provides quality and noise assessment profiles. We have Sensoscope, which is designed for the quick detection of metagenomic components in a data set. So here I'm going to show you how to generate a BCO from a computation. As we've discussed, the Biocompute object is a record of any computational analysis executed. And it's supported by the Hive platform. So to create a BCO in Hive, the BCO app can be accessed by selecting the completed computations and clicking the BCO icon from the menu bar. So we will be developing a BCO to record an alignment analysis that used Hexagon, a sequence alignment tool. And Hexagon was used to align human DNA samples from whole exome sequencing of lung squamous carcinoma patients against the human reference genome GRCH38. I ran this analysis pre-workshop for the sake of time, but on average, this alignment takes between 30 minutes to an hour in Hive. So first we'll click on the selected analysis. You can click on one or multiple. It really doesn't, it's whatever you'd like to record. You click the BCO icon here, and that will take you to the BCO app in Hive. And you first need to start with the name. This public name field allows researchers to input free text values using common biological research terminology. So I'll title this BCO, we'll say alignment of human DNA samples from whole exome sequencing of lung squamous carcinoma. Here we have the version. I'm going to version this BCO 1.0. And here I can specify a reviewer and the reviewer's contribution type, which is a choice taken from the path ontology. I'll assign my colleague Hadley King as the reviewer for this BCO, and I will list his contribution as created with, and I'm going to say that this is not yet reviewed. Right, here's a space for the Creative Commons license or any other license information. The default or recommended license can be Creative Commons Attribution 4.0 International, and that's what I'm going to be using here. And I also have to include myself as a contributor. Next, Hive will automatically populate the computational data based on the object ID input. So right here, I just want to confirm that this is the correct object ID. This is the hexagon alignment. I click Generate Information. It provides all the parametric details, all the inputs, all the outputs, the locations and the names. Then I want to click on Extra Information. Now, the BCO usability domain is used to facilitate searchability and communicate the BCO's usability. It's a space to help determine when and how the BCO can be used. So, and then I can select keywords right here. I'm going to say my keywords are alignment, hexagon, Hive hexagon, some sequencing. And then we have the namespace, which I'm going to include the external references. I'm going to include the bioproject that we pulled the data from. So the namespace is bioproject, PRGNA, and the ID of the project, 222266, and the access time, let's say today. And then here we have the external data endpoints. The service access does Hive. You need to be able to run this computation. You need to be able to, log into Hive, and that's why I'm including the URL for the login page down here. And lastly, we have the error domain, which can be used to determine what range of input returns outputs that are within the tolerance level defined in the subdomain. And it consists of two subdomains, empirical and algorithmic. The empirical error domain determines values such as limits of detectability, false positives, false negatives. It could also include, the number of alignments that your output should generate, and the algorithm. And so in this field, we're just going to have, let's say 45. The correct result should be 45 aligned sequences. This lets a user know, a researcher who's trying to replicate my analysis, know that if they get more or less than 45, this analysis did not run correctly. And then the algorithmic subdomain is descriptive of any errors that are originated by the fuzziness of the algorithms. I'm going to say not applicable here because I don't have any algorithmic error for this pipeline. And once I'm sure that I've completed all sections here, all necessary sections, I should be able to create the BCO. And I'll receive confirmation that the object was created. I'm able to download the BCO JSON or the BCO summary. So once complete, Hive has recently made some updates to improve the ease of use of BCOs in the platform. This BCO viewer in Hive works on any validated BCO that follows the BCO schema, IEEE 2791, and is stored in Hive. To view a BCO in Hive, select your JSON file and click File Preview. Using BCO Tree View to view a BCO, you will now be able to expand or collapse the BCO domains in the tree view to see or hide elements within a domain. So here I can expand provenance, I can see my reviewers, I can see my license, I can see my usability domain. So, if you'd like to experiment creating BCOs in Hive, please feel free to register here. Just go ahead and scan that QR code and you should be able to fill out registration. Any questions you can put in the chat box or ask me in the breakout room. I'll be joining Rahi in the knowledge base breakout room.
Okay. All right, well, thanks very much, Janisha and Hadley and Rahi. So we'll move into the second half now where we go into different breakout rooms. So we'll have Hadley and Janisha. So Hadley and Janisha, you guys are going to start up a separate instance. Is that the way that you want to do this? Rather than having me assign. I think when we went through this, it looked like we, the only other option was to assign people. So it might be easier to just have different people start up their own instance. And then just put the link to it right here.
Okay. Yeah, I was thinking we could go through and have people raise their hands and assign them, but what you just said seems like that could work too.
Okay.
So,
actually, well, so there's not a whole lot of people here, so I guess let's just do it that way. So let me open up.
I mean, that way people are able to return to the main lobby if they want.
Sure.
And then we can have somebody hang out here.
Yeah, I'll be hanging out here.
Okay, great. So breakout room 1, this will be Keeney Mazumder. Breakout room 2 will be Hadley King, and breakout room 3 will be Navokar Patel. Okay. I'm going to assign Hadley to that room and Janisha and Rahi to their room. All right, great. So if you'd like to join one of these 3 rooms, please let me know. And I think the way that this works is that I have to assign you to them. So there's the general, just to remind you, there's the general discussion room, kind of general ideas and overview of biocompute and sort of strategic and programmatic concerns. That's the one that Raja Mazumder and myself, Jonathon Keeney, will be in. There's the technical room for things like the code and implementation and things like that. Hadley King will be in that. That's room number 2. And then room number 3, which is implementation in biocuration and in knowledge base. That's Janisha Patel and Rahi Navokar in room 3. So these are all open. Please let me know in the, how about you just let me know in the chat, which if you'd like to be in one of the rooms, and I'll assign you to it. Or I think you might be able to just join it yourself. If you're not, just send me a message and I'll assign you to it.
No, it looks like you can only join once you've been assigned. So I was thinking, yeah, we could have people chat, or you could go through and say, you know, to join Hadley's room, the best one, you know, raise your hand now, and then, or whatever. I'll be over there.
All right, sounds good.
All right, and I'm going to end the recording at this point because I have no idea how it works with breakout rooms.
Oh yeah, so the hosts in the breakout rooms, I think, can start their own recording, I believe,
Okay.
right?